Niranjan. So to begin with, pathogens, what are pathogens? Pathogens are harmful microorganisms. It can be a bacteria, fungi, or a virus which causes illness. So when a patient has an infection, so when he visits the doctor, so the first thing which is done is syndrome identification. That is based on the symptoms, the medications are prescribed. So the next step towards it is when he is not able to diagnose, then we go for the pathogen assays. That is what is the pathogen identification. So we have many methods for detecting the pathogens. So we have culture-based, the conventional microbiology techniques, and we have molecular-based assays, and then the current next-generation sequencing technologies. So currently, NGS is gaining traction in clinical microbiology. So that's our motivation to do this research work. So what are the diagnostic problems we face, right? So when you take the consider conventional culture-based techniques, so they're you need the cultures in pure form, the isolation in the pure cultures. So the process is very tedious, it is slow and low throughput. And when you go for the molecular-based assays, so it is very limited to the set of pathogens because it requires a specific primers and probes which require the prior knowledge of the pathogens. So the above these will lead into either lack of diagnosis or delay in diagnosis. So both are problematic because if you say lack of diagnosis, so there might be a misdiagnosis which leads to the misuse of the antibiotics, which results in a major problem today in public health, that is antimicrobial drug resistance, which is again a central role of our Ph.D. work. And then we have delayed diagnosis. When there is a delay in the diagnosis, obviously the infection will spread within the community. So both are problematic. So what we need today is right diagnosis at the right time. So that's why we have chosen NGS. So why NGS? The basic principle is all of this microorganism has the genetic material. So by sequencing that, so we can identify those, and not just identify, we can also comprehensively understand the organism. So advantages of this technology, what it brings in is, we need not grow microbes in the laboratory, and a single test, that is a clinical metagenomics, where we can identify the whole microbial composition, and we have short turnaround time. So nowadays we get results within 1 day. So you can sequence the data and also get the whole analysis done. And the sequencing technology is rapidly developing, which has resulted in a reduced cost. And now we can see in a lot of laboratories today which have implemented WGS, at least to the research extent today. And for sure in the near future, we'll see these applications in the clinical diagnostics. And it does not just identify the pathogen, but it also will be able to identify the whole genetic information. That is, we can look into the AMR genes or the virulence factors which causes the pathogenicity of the organism, or how this actually evolution is taking place. So it's a whole set of information we'll be able to obtain with this analysis. So we got to know that NGS is the right technology and the future technology for in clinical microbiology. Implementation of this technology in the clinical setting will come up with some of the challenges. So the things which we are looking into is in the bioinformatics front. So these are some of the bioinformatics challenges which will be faced if we are moving this NGS into a clinical microbiology setting. So some of the challenges to mention, we have huge data which is generating in the public health or in clinical microbiology. It's not intense, it's in 10,000s or more than that. So the main bioinformatics challenge here is to analyze this whole set of data and then convert it into the clinical actionable results. So then choice of data analysis tools. So we have empty number of tools in bioinformatics which is developed. So a clinician, he cannot sit and choose what tool to use for what analysis. So it's what the research is towards now. Basically to benchmark the best tool for each of these steps and then pipeline it together. Then we have the installation and execution of these command line tools, which is again tedious in the clinical setting. So hence the need of the hour, or what we require now, is a user-friendly frameworks so that clinicians or a non-bioinformatician can directly implement that in the clinical setting. So that's the main objective of our PhD work. So this led to the development of three objectives of my PhD work. So the first objective was basically to understand how the mutations is related to the antimicrobial resistance, and by using this information, how can we classify the data? Then the next step is, this would be working good if it's infection caused by one organism, but we have infections which is caused by more than one organism, or we call it as polymicrobial infections. So in that case, a clinical metagenomic approach will come in handy. So this approach can be utilized for pathogen detection. So followed by the third objective, which is basically, so the frameworks which is developed by these two objectives are being integrated and automated in the third objective. So all of these objectives are published and the articles are mentioned below. Yeah. So coming to the first objective of my research work, that is using WGS tool for classification of resistant types. Yeah. So whole genome sequencing is basically a sequencing entire genetic intent of the microorganism. So by using this, we'll be able to identify those antibiotic resistant gene markers, so which will help us classify the data into, for example, a drug resistant, extremely drug resistant, or a multi-drug resistant. So by utilizing this information, so we have classified the data. So to demonstrate this analysis, we have utilized tuberculosis drug resistant strains from public domain. Tuberculosis is a disease caused by a bacteria called Mycobacterium tuberculosis. So what is the criteria we have considered to classify the data, right? So here we have classified our data into drug-resistant TB, multi-drug resistant TB, and XDR TB. So the drug-resistant TB is a resistant TB, which is at least resistant to either of these first-line antibiotics. And then the multi-drug resistant is the main two antibiotics which is mostly prescribed for tuberculosis is isoniazid and rifamycin. So if it is resistant to both the antibiotics, it is considered as MDR-TB. And if it is it's considered as an XDR-TB if it is resistant to both INH, RFF, and also the second-line antibiotics which is given to treat the tuberculosis. So this is the main criteria which is used for the classification method. So this is the frame methodology. For demonstrating this methodology, we have considered 70. We have screened through all the Mycobacterium tuberculosis, which is present in the public domain, and the ones which had best quality, that is, it was 74 isolates, was taken ahead to demonstrate this framework. For the variant calling, we have used best practice GATK pipeline to identify the variants in the sample. So the main uniqueness of this framework is, so we have combined all the popular database which was available at that time, which had information on tuberculosis mutational information. The reason to do this was, one, we didn't want to miss out any mutations for the identification to classify the drug types, and also to have a unified format for our mapping approach. And also one more reason is we have introduced a flag, that is, so if the mutation is identified in all the three databases, then it is given a high confidence that this mutation is more relevant. Yeah. So the basic approach here is, so whatever the variants we have identified from the samples, it is mapped to the unified database which was created, and then we have identified what all antibiotic drug class it was resistant to. So based on the criteria I explained in the previous slide, so we have scripted so that we classify the data into drug-resistant, multi-drug resistant, XDR and MDR isolates. So why do we have to do this, right? So this is what is required in the future. So if you gave that data or in the raw data, no one can understand. So finally, what a physician or a clinician would require is the whole resistome profiling, which will be the output of the previous framework. So what we basically get here is, for instance, I've demonstrated for three patient isolates. So this is 3 patients. If you visit the doctor, right, so you have all the antibiotics which is currently used against TB. So you have complete resistome profiling against that. So obviously he'll have an informed decision on which antibiotics to be used. So this is very important today because this will hugely reduce the AMR burden, because giving the wrong antibiotics is creating a lot of antimicrobial resistance, and it's also forcing, if this continues, then a lot of people will be dying not because of any diagnosis, misdiagnosis or medication, but just solely because of AMR. So understanding this whole concept is very important. So to further, this was at a genetic level. So what happens today is, so what if the resistant is against all of these antibiotics? So what people are looking at is the alternate drug targets. So to understand the alternate drug targets, we have to look at the protein level of these genes. So we considered all the important genes, drug-resistant genes in tuberculosis, and then we identified all the codon variations which is present at these regions. So we'll be able to identify some hotspot regions of mutations in these resistant genes, and it is also validated against the published articles and deposited here. So basically, why we require this is, so we know that this is a region from this codon variation to this codon variation. There's a lot of mutations happening. So what will be our drug target is those conserved regions that would be serving us as an alternate drug targets to design the drug. So this is basically to give information to the researchers that, so these are the hotspot mutations which is happening across the important drug-resistant genes in Mycobacterium tuberculosis. And these mutations were also validated against the published articles. So the conclusion of first part of the objective one is, basically this framework, we can use it to characterize the drug-resistant types and to obtain the complete resistome profiling, so the physician can make an informed decision on the antibiotics, and based on the codon variations, we will be able to identify the alternative drug target. So now we understood that mutations play a very important role in antibiotic drug resistance. So what we were interested next is, so how these mutations are accumulated in these bacteria, how these bacteria are evolving. So this led to the second part of the first objective. What we observed here is, this mutation process is usually random, but they'll always follow a specific pattern, which we call it as mutation signatures. So this mutation signatures has become quite popular in cancer genomics today. So they're used for identification of cancer types, and also even at a subtype level, we'll be able to use this information to classify. So at that time when I was reading this, I wondered why not we apply this into the pathogen genomics. So because even here it involves the mutations. Here so they will be looking into certain antibiotic pressure, the environmental pressures like with respect to UV or tobacco. So here also we have the pressures of antibiotics, which is creating this whole mutational process. So that's why when I looked into that, there was no integrated framework which was available for mutation signature analysis of bacteria. So this led to the development of this framework. So the steps are incorporated here is, so the first step we require is a variant calling file that can be obtained from the first objective by GATK pipeline. So basically we'll tell you what are the mutations present in the given sample. So the next step which we require for this analysis is mutation count matrix. So basically this will give you a mutation spectra matrix. Basically this will give you all your mutation classes, A changing to T, T changing to C. So how much percentage and proportions of these are present. In this context, we'll be taking something called as 96 base substitutions, which I'll explain later. So we'll be also considering the nucleotides next to these mutations from at 5' and 3'. So we get even further resolution in the classification. So this is the next step. So the third step is classification of base substitution, basically the mutation catalog. And then to this, we'll apply the non-negative matrix factorization. Basically, it's a data reduction. So we have thousands of whatever the mutations we identify, signatures. We are looking into the common mutation process which is happening throughout this data. So for which it's a data reduction technique. So we extract signatures and then visualization of these signatures as a relative contributions. And the cosine similarity is obtained, so we can cluster these samples based on the mutational process. So this is the framework of MuleWiz. We call this framework MuleWiz, and it is also deposited in GitHub. So basically, the input what it takes is we have also integrated the GATK pipeline. So if you give the raw FASTQ files and the reference FASTA genome, it will create the VCF file. So basically, this whole thing is written in R programming, using R programming and Python programming. And also we have used SnakeLake workflow for developing the pipeline. So basically, it's an integrated automated mutation analysis framework. So these are some of the outputs we get from this MuleWiz framework. So basically, the first one is six-type base substitution matrices. That is your transitions and transversions. A to T, T to C. So you have 6 types like that. So from purine to purine, purine to pyrimidine, so you get 6 types. So what is the proportion of these transitions and transversions which is happening in the given organism? And then, as I told, we have 96 base substitution spectrum. So this 6, whatever the classifications, right, we will also consider the 5' and 3' end of the mutation. So which will be extended to the 96 base substitution matrix to get the further resolution into the data classification. So finally, on this, we will apply the data reduction technique to derive the mutational signatures. So finally, we'll be observing these 2 mutational signatures as a relative contribution in each of the samples. So for example, in sample 1, it is just the signature 1, completely signature 1. And in certain samples, we'll be able to observe both the mutational process happening. So this is what the results we'll be getting from this output. And then finally, the clustering method. So based on this data, we'll be able to cluster so the ones which is following the similar mutational process. So this is an illustration of that. So this is for this illustration, we have used from Patrick database 963 Mycobacterium tuberculosis across the geographical locations, India, Belarus, Russia, different countries. We were just trying to understand how this bacteria is mutating across the world. Is it same or is it different? How it is happening? So basically the data what we have taken, these 10 samples were from different countries, and we have ran this data in the MuleWiz framework, and this is the first result what we got. So what we observe here is a lot of mutations which is happening is the transitions, that is G to A and C to T. So this is the major mutations which we can observe throughout the across the world in Mycobacterium tuberculosis. As I explained here, so these are the 6 basic classification which is used. So to increase the resolution of this, we'll add the 5' nucleotide and 3' nucleotide next to it. That is ACA changing to AGA. So like this, we'll get 96 type of combinations for each, which is depicted here. So this was done for, so if you observe here, so we perform in 2 methods here. So one for the complete genome, how the mutations are taking place, and then specifically to the genes, like the important genes that is for the first-line antibiotics, which is given for Mycobacterium tuberculosis. So RPOB gene and CAG gene. So what we can observe here is, so we can observe there are different genetic contents which is observed with respect to each antibiotics, right? So there's a different mutational process which is happening with respect to each antibiotic pressure. So finally, we obtained the from all the data sets, the 2 signatures were obtained, and then it was classified. So here we can see that Sweden and Mali are the data from that is clustered together. That means the mutation processes which is happening in these countries are similar. Maybe the antibiotics prescribed in those countries are same, or all those geographical conditions when they are important. Taiwan, India, Iran has a similar cluster together. So the evolution with respect to the this bacteria in these regions, the process is same. So basically, we can derive these information about evolution of bacteria. So further, we just to understand this as a whole picture. So we identified all the antibiotic resistant genes of Mycobacterium tuberculosis and run this analysis for all of the genes for understanding how the mutations are taking place. So irrespective of a lot of regions, we'll be able to see there's the same mutations which is happening across the countries in catchy gene, or in case of RPO, that is the first line antibiotics which is given, right? So irrespective of the regions, the mutations what? So that's what we can observe a specific pattern of mutations in these bacterial evolution, irrespective of where this is coming from as well. So concluding this objective 1B, so basically, mutris can be used to characterize and visualize mutational signatures in bacteria, not just in tuberculosis. It can be applied to any of the bacterial sample and even in the virus sample as well. Then it can be used for biomarker discovery. The genetic context, whatever we identify, right? So we'll be able to identify what is the genetic context which is common across the countries, across the world, right? So that can be used as a biomarker for the identification of these resistant types. So that is one advantage. And then this will also cluster the samples into similar mutational processes. So we'll be able to group the samples based on its mutational signature. So you see, these are some of the applications of mutris. So the last short bits of study from objective 1, that is amino acid substitution prediction. So we thought it would be very interesting to know how these how these mutations are happening at the protein sequence level as well. So for which first we utilized two approaches here. One is PAM approach, point accepted mutation approach, and another one is PSSM approach. So this position specific matrix. So first PAM approach will give us the overall what is the mutations which is happening at the codon level in the overall sample. Whereas the position specific, we wanted to know at what position these mutations are more. Then comparing it to, like, for example, alanine is changing to glycine. So you get an overall position of 30% it is changing across the samples. But we wanted to know how much it is changing at position 1, how much it is changing at position 2, and so on. So for which the position specific matrix was implemented. So this is the methodology which was free. So first, the input for this is a multiple sequence alignment. So you can basically all the protein sequences of RPOBG, for example, is being aligned. It is aligned using MAP program, and then a tree generation is done. A phylogenetic tree construction is done based on a distance similarity matrix. Then an amino acid count matrix is generated. So by comparing these sequences, how much changes in amino acid is taking place being calculated in the amino acid change count matrix, that is 20 cross 20 matrix. Then followed by the relative mutability of the individual amino acid. So how much each of the amino acid is changing within the given sample, and followed by the mutation probability matrix. So what is the probability of each amino acid to change in the given sample? So this followed by parallelly, we will be able to also obtain the position specific probabilities of each of the amino acid chain, which is implemented by using PSSM approach. This is just to illustrate this. Basically, we have 7 sequences. So then this will be an input. This is 7 aligned sequences, and followed by the distance matrix is created. So what is the distance between each of these sequences? That is what is the distance matrix. So based on the distance matrix, the phylogenetic tree will be created. So this will be a bit huge when we are taking a lot of samples. This is just for demonstration. Yeah. Followed by, this is amino acid count matrix. So how much amino acid is changing, right? From, for example, here, how much A is changing to another amino acid. So here it is 3 times. So that will be depicted here. The first column, 3. So followed by here, we apply a bit of mathematical formulas to calculate the probability scores of each amino acid chain. So this is the mutational probability matrix. And PSSM is similar, but it will give you the position specific matrix. So this is just a short bit of study which we extended, so which can be implemented for understanding the codon variations. Yeah, conclusion to this is what is the application of this PAM and PSSM. PAM serves as a scoring system in alignment algorithm and can be used to know which amino acids are more conserved than the others. And PSSM will give you a quantitative description of degree of sequence conservation within each position in the multiple sequence analysis. This comes to the objective one. Any questions? Continue. Yeah. So now whatever we looked at is in infections which is caused by one organism, right? So when we have infections which is caused by more than one organism, then the approach which comes in handy is clinical metagenomic approach. Yeah. So basically what is clinical metagenomics? It's an approach where you can sequence the organism without culturing them. It's an uncultured based technique, and it will identify all the pathogens which is present in the given sample. This has become quite popular in environmental samples, because if you want to identify the microbial community in a soil, in the water, or even a gut microbiome. So these are some of the applications of this metagenomics. But now, since these technologies are in handy to many clinical research now, so they are what they are forcing is they name this as MNGS technology, that is clinical metagenomic technology. So in 5 years, this will be used as in 5 or 10 years, it will be used as clinical routine diagnostics. So that's the motivation of this work. So basically, when we looked at, there are a lot of tools which is available in clinical metagenomics. So we wanted to first basically benchmark these tools, which tool will work best, and then also create an integrated pipeline. So given a patient sample data, you'll be able to identify what pathogens are present and what are its AMR profile as well. And we name this framework as MNGS. So this is the workflow which was created to do this work. So basically the input data here, what we get is the MNGS raw data, that is metagenomic sequence data. So the first step is the quality check, which is done by FastQC. Once the quality check is okay, or if it's not okay, then we'll go through Trigomatic tool to remove the reads, low quality reads. And then finally, we'll map it to the reference genome. That is, in this case, it is human genome. Because since this is a clinical data, right, so you have the traces of human data, genome data itself. Because you're not isolating the pure culture here, so you're directly isolating the DNA and then performing the sequencing. So we need to remove the host genome. So this is the host subtraction method. So when we map this against the host genome, that is human genome, or if it's any animal infection, you can map it to different genomes. So there's an input insert format which is given. Input can be given either a human genome or any other genome which you want to map. So basically, here what we are doing here, once we map into the host genome, we'll remove all the host genome, and the ones which is unmapped, right? So that is your pathogen data. So that is taken ahead for the assembly. So here we have used meta states for assembling the data. So this will basically output the FASTA file. So now this will have all the pathogens in it. So what are the pathogens which is being present in a given clinical sample will be there in this data. So first step is we look into the classification, identification of the bacteria. So what we have done here is we've used two tools. One we've used MetaQuest. It uses a different database, Silva database for 16S base to identify the pathogens. And there is another tool which is called as MetaPlan, that uses marker-based genes for identification. So we've integrated both of this. And MetaQuest will also help us, understand the assembly metrics. That is also very important. So if the assembly is wrong, then all your downstream analysis will go wrong. So basically, your assembly should go right. So followed by, once we identify these pathogens, then we are going to identify all the genes, important genes present in the pathogen. So that gives us more comprehensive information to that. So be it AMR genes or virulence factor genes, which is causing pathogenicity, and also the plasmid which is available in the given data. So these additional analysis can be done. So finally, they give us a report. One is the ITARIS report, different laboratories. So this should not be the case. So that's why there should be a standardization and the version control in the frameworks. So which we have implemented in the third objective. So when we observed in different, we have observed between two important tools, and within the tools different versions. I've put up just two of them in the presentation today, but we have performed it for all the other tools as well. So the metaplan, if you compare version 2.6 and 2.96, so you can see a lot of differences. So the previous version was able to identify the Candida species, that is fungi. But if you see the recent versions, it is not able to identify that because in the database, now they are restricted only to bacteria and archaea. So similarly, previous version was able to identify Staph aureus, but the recent version was failed to do so. And if you see in the meta-class, it is able to only identify Serratia at the species level, but not at the genus level. So these are some of the challenges we face when we are implementing bioinformatics tools. So we also identified what is the sensitivity and positive predicting values of how these tools perform. So based on these results, so the meta-class gave a good results, and even the metaplan 2 was giving a very recent results, which is implemented in our final framework. So in addition to giving the identification of organisms, so we were able to identify what are the genes which is causing resistance in these pathogens, and also profile them against the popular antibiotics which is used to treat them. So if you see the sample tree is resistant against a lot of antibiotics. So if diagnostics and also informed decisions on this will help in better treatment. So concluding this, so basically this objective 2, the critical steps in the clinical metagenomics that is being pipelined and integrated into a framework called ClinMNGS. And we have compared the various versions of tools of whatever being used in each of these steps, and the ones with the best were integrated in the final ClinMNGS framework. And in addition to identification of the pathogens, we'll be able to also comprehensively identify the AMR profiling, the virulence factors which is causing the pathogenicity of the organism, and also the plasmids which is present in these organisms. Coming to the last objective of my PhD work, so basically what we have seen in objective 1 and 2, right? So that is integrated and automated in the third objective. So the main goal for this is to provide accurate, reliable and reproducible diagnosis in clinical framework. So basic requirement what we have achieved in here is the automation of this whole workflow, the reproducibility of results by integrating all the version controls, and the scalability of samples. So it will work on either 1 sample or 10 sample or even 1,000 sample, and portability. So this workflows can be shifted from one system to other systems. So we have performed this in a laptop, we have performed it on a desktop, we have performed it in a HPC cluster as well, and it all gives the same results. So it's tested across all the platforms. So and these frameworks are written in Python R programming, and for pipeline development, we've used a Snake workflow management system. So two frameworks which has come out of this study, one we call it as Nucleus from the objective 1, and the second one is ClinMNGS. So this is the framework of the ClinMNGS work. So wherein we have integrated different process for each of the steps, and it's just a snippet of one of the process how we write the programming. And basically this whole program will output these reports. One is your quality check report, and then assembly metrics report, the Corona chart, that is the identification of all the pathogens, and then the abundance file from the metaplan, basically tell you how much relative abundance of each pathogen is present in a given sample. And finally, the AMR and virulence factor profiling. This is a complete workflow which is built, and you'll get a report like this. So if you click on the plus sign, it will also show you what versions which is being used. So user or researcher can change the versions accordingly that modification has given. And this is the runtime of the samples. This was in the cluster. And this within some seconds, you'll be able to do the entire analysis of this work. So concluding the objective 3, basically to ease these bioinformatic analysis, the workflow, the frameworks were developed. The one is Nucleus and ClinMNGS. So utilizing these pipelines will liberate users to set up these tools, installations of these tools, and also it also supports the sharing of the workflow and data. So you basically can toggle this whole workflow and can share it across the researchers and others, so they can continue working on that. So that features is made available. Then the researchers can also more extend the workflow. So for example, if he wants to add one more step, so there's an opportunity to do that. And he wants to use different versions of software, so there's an opportunity, they have given an option to change the versions of the software as well. So these are some of the outcomes of the research which is published in the publications and conference and repository in the GitHub. Future work to this is, one is we need to update and expand the databases. There will be more databases which we'll be discovering in the coming years. So basically to add them into the first objective. Then nowadays people are looking at artificial intelligence to predict mutations. I think that would be a wonderful opportunity to apply to identify the resistant mutation patterns. And then Nucleus, it was only checked for Mycobacterium tuberculosis, but we can also look into other important pathogens as well, how they behave, and also validate the ClinMNGS with more clinical data. These are the references and publications. I would like to acknowledge firstly Dr. Vidya Niranjan, my supervisor. Without her, I wouldn't have started this Ph.D. So she's been very supportive, and she's also given me so many opportunities which has increased my research horizon. She's helped me work in many places and many projects, which has made me help this understand this work much more better than I ever thought. And my family and friends, they have been great support throughout this journey. And then I have to mention these girls named Pooja and Suchitra. They are my friends and then PhD mates as well. So we have had a wonderful journey throughout together. We have had fun, we have had hard work together, and it was amazing. And these three guys, and Akshay is here, he's been my critical reviewer from the day one. So whenever I need help, I've always called him up and he's never said a no to that. And Sanjay and Vasan, so they have helped me in the initial bits of my work when I was experimenting, understanding how to do this data analysis. And then Sudarshini and Varun, they are students, MTech students. She helped me with the PAM and PSSL metrics, and Varun a bit on the understanding the metagenomics work. And then Lokesh sir is one who gave me the facility to handle the HPC. So whenever I've gone in, he's always there to provide that. And Pradeep and Shivkumar sir in the office, they've always helped me whenever I need any resources. And the TechWill for funding me initially, and Institute of Bioinformatics, where Madam helped me to work there for some time as an intern. That has initiated the knowledge of NGS inside me. So that I really want to thank them. And then KIMS was a wonderful opportunity which I got. Like last year, I could work on this global AMR surveillance project. So after PhD, I learned a lot more there. And then Queen's University Belfast, I was able to do work as a visiting research associate for 1 year. So that helped me even more technology, like work on single cell RNA sequencing, data analysis, and use their facilities also, the library. I really want to thank them. And now currently my DSC family, the Anand Sar College, so they have been very kind and supportive for my last phase of PhD. And I would like to thank one and all present here and all the staff. And I'd like to thank Manjuna sir. He's always been very helpful for me. Whatever documentation he has, I require is always provided. Thank you, sir. Yeah, I'd like to thank him a lot. Thank you.
Understand having two queries regarding your first objective and second objective. I'm just wondering to see you are mentioning a drug. Drugs are responsible for creating mutations or imposing mutations in pathogens. What is the mechanism?
There are different mechanisms which is involved. So one at the gene level because of antibiotic selective pressure. There are in certain organisms, for example in Klebsiella pneumoniae or other organisms, there is the carbapenemase, the enzyme-based activity. So there are enzymes which is produced against these antibiotics, basically to hydrolyze those antibiotics. And they're also, in case of another mechanism of drug resistance is in bacteria, we have something called efflux pumps and pores, so which will able to let these drugs inside the cell to kill it. So how the evolution is happening is the size of these pores have been reduced, so the drugs are not able to come inside the bacteria and kill it. So that's one of these are the three mechanisms.
Understand? Having a different perspective.
Okay.
Pathogen replication and all that, because DNA replication process is a major thing.
Yeah.
Where mutations get acquired.
Yeah.
So do this process is getting hampered by the drugs also?
They have the drugs also play. One is it can be by clonal expansion. So the mutations can be passed on to different generations, coming generations through the clonal expansion as well. And also if that whatever expanded, right? So now it is exposed to a lot of drugs, new drugs. So then there is an antibiotic selective pressure which is causing the mutations against those antibiotics as well.
In another word, such type of antibiotics may cause one mutation into host also. Into host also.
Yeah.
If it is causing in bacteria or pathogen, do they will cause some mutations in host cells also, some targeted mutations?
What I believe is whenever there's drug development process, so these drugs are specifically targeted against the mycobacteria, and they also look into the entire human proteome, so it doesn't target any of those proteins as well. Yeah, that is what is usually.
Just to keep bacteria.
Yeah, so it has to.
If it is creating mutations in pathogen, then definitely they have some side effects.
Correct.
This one. Second thing is we are having like first position, second and third position.
Yeah.
Which position is more targeted by the bacteria who are having this India?
So come on, next one first.
First colon position, second and third colon position.
Or first, second, and third.
So which position is more targeted? Because third position may not may not be detectable because your need and your amino acid will not change.
Yeah.
So that may not be noticed even. Or your program is capable to do that.
Oh yeah, it will be able to identify the third position.
Third position.
Yes.
Which position is more targeted?
I think the second.
Second.
Yeah.
So another last question.
Sure.
This is like a window type program.
Yeah, it's basically command line. It's not
These
GUI.
are command line.
These are command line. They're not converted into GUI.
Linux
command line based.
So can such programs be implemented in COVID like?
Yes, definitely, sir. Actually, those transitions and transversion mutations are quite looked into SARS-CoV genome, how these evolutions are happening. So that was one of our objectives, but it is not.
We already have published for Indian population about these things.
The
publication has been done. The lab has worked on that separately, and we have already published a few data on SARS. Same concept has been done.
Yeah, because we have done it already. The 50 program and find mutations.
No, actually, the program to settle in one of the system itself took some time. I requested her to come and settle. It's very difficult, and we are looking up to evolve it and make it.
I think now a lot of people are using Nextflow pipeline. So nowadays globally, so that's my next objective. I want to learn that and implement this into a Nextflow. So it's more widely used. So that's my next topic.
The same concept.
Continue with this.
And we have also cleared an AML with cancer, and 2 more people were published. The lab is already working on this.
This is a nice contribution. Means create a signature for the entire pathogens available.
Yes.
Based on that, which antibiotics may be banned or not be used.
Yeah.
Outcome can really complement with efforts and outcome adding to research. So, I congratulate you and your supervisor as well as your department and institute for having a good outcome in very precise way. Thank you very much.
Thank you very much for coming here.
Any other questions?
Yeah. Audience, there must be some questions from students, specifically masters and other PhD scholars.
From electronics, I don't know, this question is out of context.
That's fine.
So basically, I call the mutation probability. So that if many mutations happens, we have that study of how much variance is there. Like for example, if you study that some kind of patterns, the mutation happens.
Yeah.
You just take more number of samples.
Yeah.
So if you take that another kind of set, so will there be any difference in the probability, the variance? I mean to say.
Yeah, that's all. If you take it at the whole genome level, right? So then what we observe within the pathogen is it is similar. The probability of change is similar. But when we specifically look at these drug-resistant genes and specifically look at those regions, then the probability of these mutations varies. So that's what gives us information further how they are evolving or changing and how we can target that as predictions. The values, the probability scores will change.
In the probability, we say variance. Means that if anything changing randomly, how much change is happening, we have terms called variance.
Or we're talking like
quantity, how much change is there?
Like a HMM model and those states and transitions.
How much change is there in one framework? One set of study plots.
Yeah.
How much change in genome?
Overall, okay.
Do you have any?
This doesn't have it.
That is so much tricky thing because now in bacteria we are identifying organism based on 16 years also some specific signature genes. So there if you are having mutations, we are not counting entire genome to specify something as a new variant. You are targeting some genes which are taxonomically used. So there usually for strain calculation 3% is a cutoff. If more than 3% variation you will observe in certain type of genes which are highly conserved, then it is termed as a new species or maybe new strain. But this is always question science can't answer it properly.
Okay.
Because one gene may not be entire gene. This is maybe 0.1% or sometimes 0.1% of the entire gene. So good question. So I take opportunity to declare that this virus can based successfully, and whatever requirements you are having to for the requirement of the university or PhD, she has wonderfully built out the thesis as well as having very nice support with the publications on your PhD work. And so far, what presentations you had on her work is to the mark and sufficiently security. So, as an external reviewer, I don't have any question in mind. And without hesitation, I will come under the award for the award of PhD degree to Vishwesvaraya University. And I wish you good luck for your future. Continue this work. This is very much required in India. We are suffering with India like anything. Government is pouring money like anything in where this drug resistance detection labs in most of the government hospitals. Government is having a specific program. Still, they are not using such type of tools. They are using like ELISA like this.
Yeah.
It will replace many things and it will restrict the drug use in our country. So that is a bit of your work. So I compliment in total. Thank you very much.
Thank you very much, sir.
Thank you, sir. So just a few things for upcoming PhD students or people who want to do research. So one thing I want to tell about Akshatha is I was also new here and she is also new. We thought we will do PhD. Only thing we had is we have seen a lot of difference at ISC, Indian Institute of Science, where only aim I had is to have such a bio which is going to the standard of ISC, which was happening today. For that, there was a lot of things we helped because it cannot be done with only one supervisor. There must be a teamwork. So we have sent her to ISC to do those parametrics. So this should be with all the PhD students that you cannot expect anything from the supervisor only because she can give you or only certain things. Like this is my objective. We want to have some clinical thing in bioinformatics. That is what the supervisor can do and give you all the support. But any PhD student should find an opportunity because we have a lot of collaborators. Ketkar, you must be know, he's extremely good in microbiology. We have a lot of collaborations also going on. We have those things. So anybody doing PhD should have a standard, set a standard, and it cannot be done like even Akshatha when she came, because everybody think PhD is such an easy, it can be a cakewalk. It is not. To come to this level, the travel she has gone and where we have gone through as a supervisor and the team, everything is something very important. And another thing is when I see some of the PhD thesis in RBC not to have any thing, she was so, even the MTech students' thesis was exactly the same way. So this is one thing I want to congratulate Akshatha for setting a gold standard, and henceforth from biotechnology, this must be a sort of benchmark for any PhD. We will try to have it as a research committee. We will make certain things if it is not coming the level standard. She has 2 Q2 paper and 1 QT paper. That is the level of PhD. So I thank her, and as well as this PI is always a thankless job. There was many things where I pushed her a lot to do this, but finally today I'm happy. This is as good as any of the IIC defense, which I know whoever all from IIC would have seen that this is the level of thesis which either my PhD students or anybody in biotechnology the students should have. So congratulations, Akshatha, and thanks for not either they have to come this level or let them leave it. If the system is not supporting them, so you should either find the best or leave it. PhD is nothing. You can get a job without PhD also. So anybody, since MTech is also there, if you want to do PhD, because yesterday principal was telling he was interviewing 50 PhDs for the postdoc, and he found they are not eligible for as a PhD. He didn't take anybody. So it's not only completing PhD, it is giving a standard PhD which will lifelong. I was thinking, since I have done an IOB with Akhilesh Pandey, anywhere that we go, we will know that how to do research, how to help. So this is something I'm telling everybody, right from the department or upcoming PhD students, have a standard, gold standard and work for it. Don't go for compromise. So that is something I want to take this occasion for upcoming PhD students and also the supervisors to have a gold standard. And because the college is not compelling anybody to have PhD student. If you have a PhD student, make sure, maybe you are not at the gold standard, but you can set a gold standard for the next people to come. So this is my thing for all of you. Thank you.